All right, so have you ever like looked down at the ground and, wondered what's actually going on down there? I mean, it's dirt, right? But today we're going like way deeper into the hidden world of soil, all through the lens of a microscope. And it all started with this snippet you sent us of a conversation between Kesser and Steen at a garden center.
Yeah.
It turns out this super basic question, like, isn't soil just soil?
No.
It opens up this crazy landscape. Especially for someone like Kesser, who's really into all those tiny little life forms that live in it.
Yeah, it is amazing how, just that initial kind of spark of curiosity that Kesser has, about all those microscopic things in the soil can lead us to explore these complex and vital ecosystems. And, that's kind of what this conversation highlights, that desire to understand all this stuff that's going on right there under our feet in our gardens.
Right, exactly. So for this deep dive, we're on a mission to give you a clearer understanding of why different soil types even matter in the first place. And if you're feeling adventurous, you know how you might even collect your own soil samples like Kesser, and then peek at all those amazing bacteria and fungi and the other microorganisms calling your garden home.
Right.
And we'll also touch on what the presence or absence of, certain organisms might tell you about your soil's health.
Okay.
And we'll tackle a common misconception about, prepping soil samples for, this kind of exploration.
Great. Looking forward to it.
Yeah, me too. This is going to be fun.
Yeah.
So, we think about this in the big picture, right? Understanding your soil at a microscopic level, much like Kesser's trying to do, it can totally transform how you think about gardening in general.
Yeah.
And all the fundamental processes that make plant life, possible. It's not just dirt. It's like a whole living ecosystem.
Absolutely, yeah. It really changes your perspective.
Okay, so let's jump into this garden center conversation you sent us. Where were they?
Yeah, they were just surrounded by, all those bags of soil, rose soil and tomato grow bags and top dressing, all those different specialized mixes.
Right, yeah.
And Kesser was trying to explain to Steen that it wasn't just dirt, that there was a difference.
And Steen was coming at it from a more practical angle at first.
Yeah, exactly. I just take soil from different parts of the garden and, I mix it all up.
Right, just dirt is dirt.
Exactly, yeah. He even talks about how those different parts, the little stone, sand, humus, clay, all that stuff, it has different densities. So even though they look the same to the naked eye, Kesser obviously wants to dive deeper into the microscopic level.
Yeah, he's all about the tiny stuff.
Right. And Steen's point about the density is really interesting because it kind of touches on that whole challenge of getting a truly representative soil sample.
Right.
Especially if you're trying to quantify, measure all those different components. Even a teeny tiny little bit of soil can have vastly different proportions of all that stuff.
Yeah, that makes sense.
So that's why Kesser's point of looking at the microbes becomes even more interesting because, they're distributed throughout that whole matrix.
It's like this hidden world we can't see.
Totally.
Okay, so let's talk about this challenge of quantitative versus qualitative sampling, right? Especially because Kesser wants to see what's living in the soil.
Right.
Kesser explains to Steen that even with super precise tools, it's really tricky to take a consistent volume of all those different soil components.
Yeah.
Because of things like, varying water content and stuff.
Yeah, and this is a key difference between, doing rigorous scientific analysis where you need to know the exact amount of everything, and when you're just looking at the organisms under a microscope like Kesser. The goal is not to measure and count everything. It's more about just understanding what's there and maybe what it's doing.
And Kesser even brings up this concept of a standard method.
Right.
Like how their professor, Mr. Hendricks, taught them. The main takeaway here is that if you want to compare what you see with what other people are seeing, or even your own observations over time, you got to follow those established protocols.
Exactly.
Otherwise, it's just apples and oranges,
Totally. And that's a fundamental principle of any scientific endeavor. It's all about reproducibility, making sure everyone's using the same methods so we can compare and share findings,
Yeah, it's like speaking the same language.
Exactly. Even if you're just doing home microscopy, Kesser having consistent methods will make your observations more meaningful in the long run.
Yeah, so it's not just a random look-see.
Right. It's more systematic.
Okay, so going back to that garden center chat, what happens next?
Well, Kesser brings up these old writings that they found in a barn, and it's kind of interesting because it shows that even way back then, people understood that there were differences between soil types. They might not have had microscopes, but they could see how those different soils affected how plants grew, So it's not a new idea. It's just that now we have the tools to understand the details.
Right.
And I think that's what Kesser's trying to get at, that soil isn't the whole story.
There's way more to it than meets the eye.
Exactly. And then Steen kind of shifts the conversation.
Okay.
He starts talking about microbiology practices and stuff like dilution and using scientific notation, like 75 times 10 to the fourth instead of 750,000.
Oh, yeah, right. And that whole concept of less than 1 instead of 0 when you're counting microorganisms. Why is that important?
Well, it's all about how microbiologists deal with the massive amounts of microorganisms in a sample. Dilution helps to break those numbers down so they're easier to work with, and scientific notation is just a shorter way of writing those huge numbers.
So 75 times 10 to the fourth just means 75 followed by 4 zeros.
Right.
750,000.
That makes sense. And the less than 1 thing is important because even if you don't see a particular microorganism in a small sample, it doesn't mean it's not there at all. It could just be that its concentration is so low that it's below the detection limit of your method.
It's like you just can't see it yet.
Exactly.
But that brings us to a key point that Kesser makes. When you're observing your garden soil at home under a microscope, like Kesser's planning to do, it's not about counting every single microbe.
Right.
It's more about figuring out what types of bacteria and fungi and little animals are present and, maybe what's missing.
Yeah, and that shift in focus from counting to identifying is a big deal for people who are just starting out with microscopy at home. You're not trying to get super precise numbers. You just want to understand the composition of your soil's living community, and maybe what they're up to.
It's like taking a census.
Exactly.
And Kesser uses a great analogy to explain this, comparing the soil microbiome to the human microbiome, like all those bacteria in our gut and on our skin.
Yeah, it's a fantastic way to picture all that unseen activity happening right beneath our feet.
Right. So understanding your soil's microbiome isn't just like an interesting fact. It's actually like a key to having a thriving garden.
Absolutely.
Like how a healthy gut is important for us.
Yeah, a healthy microbiome in the soil can lead to stronger plants, better nutrient absorption, and they'll be more resistant to diseases, which means you don't have to rely on fertilizers and pesticides as much.
It's like giving your plants a natural boost.
Exactly.
And get this, Kesser says plants actually use bacteria in their roots to take up nutrients.
Yeah, isn't that wild?
It's like this whole unseen world working together to make our plants grow.
Yeah, it's mind-blowing when you really think about it.
So, while Steen might see just dirt, Kesser sees this whole bustling city of microscopic activity.
A microscopic metropolis.
Right, and then they talk about how to actually manage this microbiome.
Okay.
Kesser compares it to taking probiotics. Which is funny because I was just thinking about gut health. Right. And it makes sense because just like with probiotics, you can't just throw those beneficial microbes into the soil and expect them to stick around.
They need the right environment.
Exactly. It's like setting up a nice home for them.
Right. Providing food and shelter.
So Kesser emphasizes that good gardening practices like composting and mulching and watering the right way and using cover crops, they're all essentially like giving those microbes what they need to thrive.
Absolutely. It's about creating a diverse and healthy soil environment.
And while you can buy and introduce microbes, Kesser says that your gardening habits are what really determine whether they survive and actually do anything good for the soil.
It's true. You're basically cultivating a whole habitat down there. It's like introducing fish to a polluted pond. You can't expect them to thrive if the environment's not right.
So how do you know if the environment is right?
Well, that's where observation comes in.
Okay.
Kesser sends a quick message and gets a response about these standardized guidelines for soil sampling for microscopy. And it turns out there are well-accepted ways to collect those samples if you want to actually see those little guys under a microscope.
Okay, so is this something that anyone can do at home?
Yeah. It's definitely not as strict as a clinical setting, but following some basic guidelines can make a big difference.
Gotcha. So what are those guidelines?
Well, there are a few different methods that Kesser mentions. First, you've got the direct wet mount, which is basically just mixing some soil with water on a slide. It's super simple, and it's great for seeing bacteria, protozoa, fungal spores, and amoebae.
So it's like a quick and dirty way to see what's living in your soil.
Yeah. Exactly. And then there are some staining techniques that you can use, like gram stain for bacteria or lactophenol cotton blue for fungi.
Okay.
These are like special dyes that make certain structures easier to see.
Right. So it's like highlighting the interesting parts.
Exactly.
And then there's something called culture-based enrichment, where you kind of give certain organisms a little boost so they're easier to spot under the microscope.
Yeah. And then there's a really interesting method called the soil food web methods.
Okay.
It's based on Elanium's protocols, and it focuses on observing bacteria, fungi, protozoa, and nematodes all together.
So it's like looking at the whole ecosystem in action.
Right. It gives you a more holistic view of what's happening in the soil.
And Kesser points out that this method isn't about getting super precise numbers.
Mm-hmm.
It's about getting a representative living snapshot of the soil, like who's active,
Yeah.
how diverse it is, and if there's like a good balance between different groups.
So it's more about observation and less about counting.
Right.
And that's really important for home enthusiasts because you're just trying to get a feel for what's going on down there.
You're not trying to write a scientific paper.
Right.
So Kesser also mentions that small differences in things like sample weight or soil density won't really change what you see under the microscope.
Right. It's all about the big picture.
Okay, so now we come to a big warning about drying soil before analysis.
Yeah.
Kesser says drying a soil sample at 120 degrees Celsius for 14 hours is like the standard way to figure out the dry weight,
Yeah.
which is super important for things like calculating microbial biomass or nutrient concentration.
Right. It's all about standardization.
But here's the thing, and this is huge.
Okay.
If you do that before trying to look at anything under a microscope, you're basically killing everything.
Yeah, it's like nuking the soil.
So all those bacteria, fungi, protozoa, amoebae, gone.
Yeah, kaput.
So for Kesser, drying the soil is like the opposite of what he wants to do.
Yeah, it's a complete no-no for this type of microscopy.
Because the whole point is to see those things alive and moving and doing their thing.
Right. It's about observing life in action.
So drying is okay if you're doing chemical analysis or you need to know the dry weight, but it totally destroys the biological information that Kesser's trying to see with his microscope.
It's like trying to study a forest after a wildfire.
Right.
You're missing the whole point.
So basically drying is good for some things, but it's totally useless and actually harmful if you're trying to look at living things.
Exactly. Choose the right method based on what you're trying to learn.
Okay, so back to that garden center.
Mm-hmm.
What happens at the end of the conversation?
Well, Steen kind of jokingly asks Kesser if he's saving up for a laser microscope after all this talk about the details.
Mm-hmm.
And then a tin can gets caught in a rat trap.
Oh.
Yeah, it's kind of a funny ending.
Sounds like a typical day at the garden center.
Yeah. But it also shows how even something as simple as soil can lead to these really complex and interesting questions.
Yeah, that's what we're all about here.
Exactly. Exploring the world around us and asking those questions.
So to recap, we've learned that different soils have different properties, and they support all sorts of different microbial communities. And if you're interested in seeing those microbes for yourself with a microscope, the way you collect and prepare your sample is super important.
Absolutely.
And the goal is to get a living snapshot of that soil. So don't go drying it out or anything like that.
Right. Keep it alive.
Because ultimately, understanding that living component of the soil can give you a whole new appreciation for how everything works down there.
It's a whole hidden world that's supporting our gardens.
And hopefully this deep dive into the microscopic world of soil has given you some new things to think about.
Yeah, and maybe even inspired you to get a microscope and explore for yourself.
That'd be awesome.
It would. And it raises another interesting question. If the health of our gardens and even our own bodies is so closely connected to these microscopic communities, what other unseen worlds are out there influencing our lives in ways we don't even realize?
That's a great question to ponder.
It is.
Yeah.
What might we discover if we just looked a little closer?
Ooh, that's something to think about. Thanks for joining me on this deep dive.
Anytime. It's been fun.
Me too.
Until next time.